Okay, continuing with our DNA, we're going to talk about how can you make copies of DNA, because our DNA has been passed on. It's in every one of our cells. Well, how the heck does that happen? So DNA replicates itself, okay? So what happens is the nitrogen base on one strand, it's going to form 2 or 3 hydrogen bonds. It depends on if it's a double ring or a single ring, okay? So the base pairs, again, we said was A always goes with T, adenine always goes with thymine, and guanine always goes with cytosine. So there are 2 hydrogen bonds between A and T, and there's 3 between G and C. Maybe that's how they recognize each other. You know, that's something they've looked at. But again, these bonds, hydrogen bonds will hold the whole molecule together. But when it's time to make more, what'll happen is, you know, those 2 strands are held together. The hydrogen bonding will be broken down. It'll be, you know, open up the 2. So you have this twisted ladder, which is 2 sides. It'll straighten out, and now you'll have your bases. The ladder, basically, it breaks down the middle. If you took a ladder and you cut it all the way down the middle, that's what happens with the DNA. Okay? So when it's time to make more DNA, that's what it does. It opens up, and so you have your bases sticking out, and now you have new nucleotides come in and cover those bases. They match up, A, T, C, G, and it does it on both sides. So when you're done, where you used to have 1 strand of DNA, now you have 2 exact copies of DNA. So when the cell's ready to divide, now they get the same DNA. Okay? So again, when the DNA is on the chromosomes, you guys, I'm pretty sure know what chromosomes are. Okay, there's special proteins with the chromosomes. They're called histones, and they make the DNA coil up in the chromosomes. So you can actually see it, like when a cell's getting ready to divide. If you look at it under a regular microscope, you can see chromosomes when the cell's ready to divide. Okay, so again, those histones, they're like proteins that they curl around. Okay, it would be like if you took a string and curled it around your finger. You can make it in use a much smaller area. Okay, your finger would be like the histone, and then you're curling up the DNA, you're curling up the string. Okay, your complete DNA sequence is called a genome. Okay, and the weird thing about your DNA is only about 2% of your DNA codes for anything. They call it, you could call it junk DNA. There's all kinds of things they can call it, but basically, only about 2% of all your DNA has information to build you, which is kind of crazy if you think about it, because there's a lot of DNA, but only a small portion of it is actually used. Okay, they actually, what they did was they broke up all the chromosomes into teams, scientist teams all over the world. They started the Human Genome Project, and basically they mapped out every chromosome. So one team might have had chromosome 1, and so they figured out, you know, this gene is here and on this part of the chromosome here, and they, like I said, they spread the work out all over, and they mapped out every single one of the chromosomes, so they can tell you, if you look at them, you can, there's pictures of it online and all that, but it'll tell you like hair color is on this gene, and genetic disorders are in these genes, and it'll tell you all of it. They mapped it all out. So they call that the Human Genome Project, and they can tell you on every chromosome what all the genes are. Okay? So if we're going to make a copy of DNA, like I was saying, it has to open up. Okay? And it starts to replicate itself. So you can see in this how these are open. We have, I think it's CCC. So it's going to look for GGG to come in. I think this is C on this side. It's kind of hard to tell. But what's going to happen is a nucleotide is going to come in with a C here and here and here and add to this. And on this side, the same thing's going to happen. So where you had 1 strand before, when it starts to open up and the nucleotides come in, you're going to end up with 2 strands. And the 2 strands are going to be the same as the one that it came from. But you're just going to have 2 copies where you only had 1. And this is the 5 to 3 I was talking about. It's just the carbon that it starts on. So like this side is 3, this side's 5. Okay, that's the primers or the carbon. Okay, so DNA replication requires several enzymes, and I have a video for you to watch. It'll talk more about those. But basically what it does is it comes in, it starts unwinding it, and then breaking those hydrogen bonds in between the bases and opening it up. And then new nucleotides come in, and then, like I said, where you had 1, now you have 2. And the 2 are the same as the one that it came from. Okay, that's called replication. Both strands are copied at the same time from the 5 to 3 direction. So it starts, let me see if you can see. the 5 to 3 direction from here to here. So it opens up and then it copies on this side, then it copies on that side, then it copies on this side, then it copies on that side. Okay? So they're simultaneously copied in that direction. And there's something called a DNA polymers that helps open that up. There's some other ones that do it, but they call that the replication fork. So it doesn't just all just break apart. It goes in small units at a time. Okay? And again, it goes from the 5 to the 3. So the 5th carbon to the 3rd. That's not important, you guys. The thing to remember is the DNA is going to open up and it's just going to copy itself. And by doing that, it brings in nucleotides to match up the nitrogen bases. There's the lagging strand, they call it. If it's synthesized discontinuous, in other words, if they're fragments, they actually have stuff that comes in and fixes that too. So, because you don't want it to break apart, okay? If you miss one of those bases, there's going to be a problem, okay? So there's a helicase, which ASE, it's an enzyme, unwinds the strands for replication. And then you got polymers that comes in and helps. And you can look, 100 nucleotides per second. And its error rate is 1 in a 10,000. It's amazing. And then they have other things that go back and read it, like proofread it, and like, oops, you missed right here. Let's fix that. So this is such a cool thing because it has stuff that comes in and makes it unwind, and it starts making copies of it. And then if it makes a mistake, they actually have like proofreaders that go in and fix mistakes. Okay? After proofreading, you can see the error rate is less than 1 per billion. Okay, so pretty good system that we have going on in our cells and we didn't even know, right? Okay, and if you have a natural mistake that occurs in those, then it could be a mutation. Now, mutations can be good or bad. Sometimes mutations are so bad the individual won't survive. Mutations can be tumor forming, cancer. Mutations can be a birthmark. Okay, so there's all kinds of things. Mutations can be good or bad. And again, a mutation is a change in the DNA, a change in that code. Okay. All right, so the role of DNA and RNA in protein synthesis is very important that we have that DNA, because what the DNA is the blueprint to build you. It's all the genetic information to build proteins. And that's what you are. You're a bundle of different proteins. We talked about proteins last chapter. There's over 1 million different proteins. Well, where do we get the information to build them? From the DNA. The DNA is the blueprint, okay? It's stored in the vault. It's stored in the nucleus. When we need that information, we go in the vault, we go in the nucleus, we make a copy of what we need, and then we leave. And then we take that information and we use it to build a protein, okay? So the way it works is the information is in DNA, okay? It's transferred to messenger RNA, and then it makes a protein. And that's pretty much everything that molecular biology is kind of built on. Those of you, if you've had a COVID vaccine, they pick up the vaccine right here, okay? Because what's going to happen is your body's going to make messenger RNA, and then it codes for a protein. So when you got the vaccine, they gave you the messenger RNA. I know I've heard of some people worried about the vaccine because it's going to mess with their DNA. It doesn't go near your DNA. It doesn't go into the nucleus. It stays away from the vault. It's already got the information. Okay, we're putting information, when you get a vaccine, you're getting the information already made. It's in the messenger RNA, so it's going to go in and it's going to start making that protein to protect you from the COVID virus. Okay, so those of you that are worried about that, it doesn't go near your DNA. I promise. I was worried about that too when they started talking the vaccine. Then I looked at what they did and was like, oh, that makes sense. Don't mess with my DNA. Already give me the messenger RNA with the code, and my body will start making the protein. Okay? And the thing with RNA, DNA, there's one form. That's it. Double helix, got your nucleotides, sugar phosphate base, that kind of stuff. RNA, there's actually 3 forms. Okay? And again, remember the difference between DNA and RNA is DNA is deoxyribose. It's a different sugar than RNA. And DNA is double-stranded, RNA is single-stranded. And we actually have 3 different kinds. DNA is the information, RNA does the work. So DNA, if you were thinking like a business, DNA would be like the boss. He's the one with all the ideas. The RNA is the one that does all the work. Those are the factory workers, okay? So RNA, you have 3 different kinds. Okay, when you make a copy of the DNA, it's messenger RNA. That's mRNA. Okay, so it's a single strand and it's a copy of the DNA. It makes a copy and then leaves the nucleus and goes out into the cytoplasm. Then it needs like an anchor. Ribosomal RNA is kind of what anchors it down. And then you have transfer RNA, which has amino acids attached to it. And their job is to bring the amino acids up to the messenger RNA, and then you put them all together and you get your proteins. And I'll talk more about that later. But that's the start. Let me see. Okay. Again, ribosomes, part of the cell. They're complexes of protein and RNA. And again, they're scattered throughout the cytoplasm, but they help with the process. Sometimes they anchor it down. Sometimes they're like the one that goes down and reads it. And there's other things I'll show you that'll probably make more sense. But again, these are nucleic acids. They're made out of nucleotides. And they're pretty important because they pretty much make us. That's the structure that builds us. That's the nucleic acids. So we're going to quit right here, and we'll probably finish it on the next part.